Yeah. Dear teachers and students, my name is YangYangYu, come from Stanford University, from China. It's a great honor to have an opportunity to share my work with you. My research title is the Dynamic Transitions in Neuronal Network Firing Sustained by Abnormal Exercise Feedback, and I will take epilepsy as an example. And my study structure is Green Professor, and my email is here. My speech consists of 3 parts, including research background, abnormal exercise feedback-induced epilepsy or periodic epilepsy. Overall, epilepsy is one of the most common neurological diseases affecting nearly 70 million people worldwide. Epilepsy fever is abnormal synchronous firing of neuronal population. Because of the imbalance between excitability and inhibition, just like the left figure shows. In that figure, ovals represent excitatory neurons and these triangles represent inhibitory neurons. On the whole, inhibitory neurons cannot effectively inhibit the firing of excitatory neurons and causing excitatory neurons to be overexcited and to show epileptic firing. Before, people think the imbalance between excitability and inhibition caused by neurons themselves, but in fact, there are many cells also making effect, that is, exercise. We can see the right figure. From the right figure, we can see the astrocyte that is astrocyte, and they closely connected with the neurons. We can look at the yellow part is the neurons. Now we know astrocyte has a critical effect on the balance of neuronal excitatory and inhibition. Not just play a structural sport, transport energy for neurons. We said astrocyte closely connected with the neurons, not only in structure and in functional. We can know from the left figure. The left figure shows that astrocytes can respond to the stimulation of neuronal activity, and we can there the presynaptic neurons can release the glutamate to postsynaptic neuron and to the astrocyte, and then astrocyte can release neurotransmitters uh, to uh, to regulate uh, presynaptic neuron involves, and that is astrocyte feedback. We also use the tripartite synapses to describe the two-way action. Tripartite synapse is a basic unit to describe neuron neuron and astrocytes the interaction, and trabecal synapse is the basic unit in neural modeling. We said exercise has an important effect on neuron firing. We can think what effect of abnormal exercise feedback can make. Before, many researchers have studied abnormal exercise function can induce epilepsy, just like the right figure shows. From the figure, we can know uh, epileptic activity can promote the exercise to make some change, like process wiring and shrinkage, and involves exercise can promote epileptic activity. and many physiology experiments verify abnormal structure also can make abnormal feedback, and few people use modern methods to study that. So we construct a feedback model to investigate the effect. The upper left figure is a functional neuron network and astrocyte network capping model. The model is raised by Emery. And the bottom left figure describes the physiology capping network of neurons and astrocytes. And from the figure, we know the orange is under the orange represent the body of the neuron, and the others are astrocytes. And from the figure, we can know in the actual brain, a single astrocyte can contact with many neurons, and neurons can be surrounded by different astrocytes. Thereby, they can forming a very complex capping network. And based on the physiology model and the Emily's model, we can structure an out-of feedback model. The model consists of 15 pre-model neurons and 15 interneurons and 15 astrocytes. In the bottom right, this formula represents astrocyte feedback, and the lambda 1 and the lambda 2 represent the feedback intensity from astrocyte to pyramidal neurons and interneurons. And the probability 1 and the probability 2 are the probability of an astrocyte successfully connecting with the pyramidal neuron and interneuron. The connect probability P is our main project. These three figures are the synchronization states of neural network change with the probability P and the lambda 2. The rho is synchronization index. From figures we know the neuron population's synchronization. This decrease first and then increase. This indicates that the appropriate astrocyte regulation can suppress the failing synchronization of neural population. But when astrocytes are excessively involved in the regulation of neuron fairing, the fairing synchronization of neural population will increase sharply. That indicates that astrocyte feedback can change neuron population's synchronization and over synchronization neuron population. That will make neural population to induce epilepsy. In this part, we studied the transitions in neural network fairing sustained by abnormal astrocyte feedback. The figure are fairing pattern of neural population, and the horizontal axis represent the time, and the vertical axis is the neuron's location. We can know from the upper figure, this picture, to middle figure, we can see the neuronal populations fairing transitions from a synchronized fairing state, just like this picture shows, and to change it to asynchronous fairing. First, the probability to equal to 0 from 0 to 0.35, and then the rule continues to increase until probability to equal 1. And in that time, the feed-like firing is more severe and synchronized. Eventually, the neuron population's firing transitions from asynchronous firing to seed-like synchronous firing. Just like the bottom picture shows, the yellow strip area indicates that the neuron population is in a seed-like synchronous firing state, and then and later it resumes to normal firing, just like the first picture. The above results indicate astrocyte feedback can induce the transition in neuron population synchronous firing activity, and review abnormal structure can induce epileptic seizures. And in some cases, epilepsy is recurring. This is periodic epilepsy. In the above study, the epilepsy has a single cycle, but we can study the periodic epilepsy with our model. The red figure are firing pattern of neuronal population. From the two figures, we can see when lambda 1 equal to epilepsy have single cycle in total 200 seconds. And when lambda 1 increased to 2.3, epilepsy from single cycle transitions to periodic. There are 4 cycles. The period change, and the left are single neurons firing diagram, which shows the 25th neuron of the population. And from that, we can see the periodic of epilepsy firing in detail. In this part, from the right figures, we can see when lambda 1 equal to 2.3, epilepsy have 4 cycles. But when lambda 1 is increased to 2.8, the period number is increased to 5, showing that the number of cycles increases as lambda 1 increases. The left figure is two-parameter graph. Red represents the periodic epilepsy firing, and black represents the periodic epilepsy. From the left figure, we know when lambda 1 bigger than 2.3, there is periodic epilepsy, and has nothing to do with lambda 2. The above results indicate lambda 1 is the main parameter to change the periodic of epilepsy. Or the other words, the astrocyte feedback to pyramidal neurons can directly induce periodic epilepsy, and abnormal feedback can change neuronal firing pattern. And above is the main content of my report. Welcome to question. Anyone can contact me with email if you have interest or question. Thank you. That's all.